Good morning, everyone. It's still morning. We have an hour of morning ahead of us. Welcome, everybody, to this incredible Institute for Human Virology 18th Annual Marlene and Stuart Greenebaum Lecture. We really appreciate 18 years of support from the Greenebaum family, who also, of course, have helped support and build the University of Maryland Greenebaum Comprehensive Cancer Center. And if you had trouble driving in today because of the construction of the big tower going up, that's the new home for the Greenebaum Comprehensive Cancer Center. So we're really thrilled about that. And the leadership of Tofika Wanikoko, another Nigerian superstar who is leading our cancer center. I'm very excited about his work. So this year we have two lecturers that are going to be speaking to us. We have Dr. Anu Osinusi and Dr. Moupall Das. Did I get that right? And we're really delighted to have them visiting us. Um, First of all, I just want to talk about Dr. Osinusi very briefly, and then we'll hear a formal introduction from Dr. Katakuzi. So Dr. Inosi leads the non-HIV virology portfolio at Gilead. She led the remdesivir effort for COVID and helped forward the first antiviral therapies for hepatitis Delta. She was actually a fellow with us here and at the NIH and worked with Sean Kotelul and Dr. Henry Mazur before joining Gilead. And I just want to comment that I'm incredibly proud of Gilead and what it's done historically in the fight against hepatitis C, the fight against COVID-19, and now the transformative impact on the development of long-acting preventive therapies for HIV. And I'm also very proud that this company always had a very forward-looking effort at trying to bring new therapeutics to the entire world and creating cost structures to support that development. And it's something that I think the company really led all other companies in that effort. And that effort has saved millions and millions of lives. I'm also very proud of the leadership and partnership that we've had at Gilead with both the NIH and Dr. Mazur, and also with Sarah Katakuzi and Aleda Rosenthal, who over the years partnered on the first in human trials of new hepatitis C therapies and bringing these therapies to very vulnerable populations in Washington and Baltimore, populations that typically wouldn't receive these cutting edge therapies at the beginning. So I'd really appreciate Dr. Kotelul's leadership in that historic partnership. We're also very excited and honored to have Dr. Das here, who's helped develop Lenacapavir. This is a long-acting HIV medication. It was named Molecule of the Year by Science, and this is a medication that completely prevents HIV infection, a transformative medication. And again, I understand there's been a recent deal to bring that medication to Africa and across the world. We think this will be a tremendous benefit for our patients in the DMV and in Baltimore in particular. So we're very appreciative of the leadership of Gilead and science. And I can't help but mention in this time of cynicism surrounding global health, surrounding vaccinology, surrounding RNA technologies, surrounding science, that you can't help but note that NIH fundamental science really forms the backbone of 99% of all FDA-approved drugs in the last 25 years and have advanced success of America. 4% of the GDP of America, 4% of the best jobs derive from academic medical centers like ours right here in Baltimore. And a statistic I learned recently from the 2025 Economic Impact Analysis of Baltimore, our academic medical center, which is 4 hospitals in Baltimore, Umroy, Washington Hospital, UMMC right next to us here, and Midtown campus, the University of Maryland Baltimore campus where we sit, our 1,400 faculty practice, which of course Dr. Kotelul leads the infectious disease pieces of that practice, and all of the providers, students, that entire academic medical center, we contribute 26,000 jobs to Baltimore alone, $5 billion in economic impact, and $2 billion in research expenditures. So we're thrilled about partnerships with Gilead moving forward. We are very working hard to adapt to challenges around funding of science, and part of that is developing rapid activation of clinical trials. And we're looking to form platform partnerships with industry, and we'd love to take our history with Gilead forward in that domain. So Dr. Kodalo did not want me to do that sales job, but I did, and I welcome our partnership. And let me introduce, I think, Dr. Catacuzzi up here.
Good morning, everyone. It's my pleasure to introduce Dr. Anu Osinusi. Dr. Osinusi is Vice President, Clinical Development, and Head of Hepatitis, Respiratory, and Emerging Viruses at Gilead Sciences. Dr. Osinusi earned her medical degree from the University of Ibadan, a Master of Public Health from the Harvard School of Public Health, completed residency at Rush University Medical Center in Chicago, and an infectious diseases fellowship here at the University of Maryland. Woohoo! Prior to joining Gilead, Anu served as clinical director of the hepatitis branch for the DC Partnership for HIV/AIDS Progress, as mentioned, a collaboration between the NIH and the DC Department of Health. In this role, Dr. Osinusi helped establish community-based research trials in underserved populations in Washington, DC, leading clinical trials that contributed to the approvals of transformative hepatitis C therapies and expanding research participation and access to treatment through innovative approaches. In her current role as Vice President of Clinical Development at Gilead Sciences, Dr. Osinusi leads a broad portfolio of programs across viral hepatitis, respiratory, pandemic, and emerging viruses at Gilead Sciences. Passionate about rapidly advancing innovative therapies, she has led the development programs for several transformative medicines, including curative treatments for hepatitis C, the first approved antiviral for COVID, and the first approved therapy for hepatitis D. Finally, Dr. Osinusi has authored more than 80 scientific publications, serves on the board of directors of the Gilead Foundation, and is scientific chair of the Sickle Cell Hope Alive Foundation. And on a personal note, Dr. Osinusi remains a trusted mentor and friend to those who have had the honor of working with her. After her recruitment to Gilead Sciences, Dr. Osinusi was the one who recruited me to the PFAP program, completely changing the trajectory of my life. Her words were, "You'll really like the work and working for Sham." And here I am 12 years later. So for all those reasons, it is my deepest honor to welcome a friend, a mentor, an incredible physician scientist, Dr. Anu Osinusi. Okay. Hello, everyone. And thank you, Sarah, for that really kind introduction. So good day, everyone. Like, it really warms my heart to be here, honestly. Like, as I look around the room, I see so many familiar faces. I see a lot of my teachers and some of my attendings. I see colleagues. I see my first attending sitting right there. I see a lot of my colleagues, co-fellows, friends from IHV, from University of Maryland, from the NIH, from DC PFAP, and I see a few Gilead faces in the room as well. So for me, this place, University of Maryland, IHV, NIH really shaped the foundation of my career, and to have the opportunity to come back and share how that's impacted my path is profoundly meaningful to me. So I'm going to start out with a couple of stories. So my journey with Maryland and IHV actually started in 2003. It was 22 years ago, thinking about it. So that was right after I'd completed my MPH. And so that year, I actually had the opportunity to work on a few bold initiatives that actually challenged what many people thought were possible. Anthony, I see you right there. And one of them was the JOCS initiative, which was actually expanding HIV treatment and care in underserved communities in Baltimore. And then I also had the opportunity to work alongside Dr. Anthony Amoroso over there and Dr. Redfield in writing
some of the first PEPFAR grants to expand HIV treatment and care in 9 African and Caribbean countries. And to many people, I see many faces in this room who've worked on the PEPFAR grants over the years. You know, those were very bold initiatives. And thank you for all the amazing work you've done that has not only transformed health systems in many of those countries, including Nigeria, where I'm from, but also actually saved millions of lives. So thank you for all that you do. And I think for me, what struck me back then about Maryland and still does today is the deep commitment to patients. You know, whether it was a patient in Evelyn Jordan Clinic or the small clinic in Rwanda that people were in, the work was very scientific, but it was also very human. It was about real people, it's about patients, it's about their families, and it's also about the community. And I also remember some sage advice from Dr. Amoroso. I told you I was going to say this. And, you know, he said to me then, like, Anu, your career doesn't have to follow a straight path. You know, just stay open to opportunity. In the Anthony way, it'll all come together. And I think that advice still remains with me today, that a fulfilling career is not about a fixed path. It's really about staying open and guided by purpose, even when that means really taking unexpected turns. And so after residency in Chicago, as Sarah said, I returned to University of Maryland for my fellowship, wonderful memories, and got to work with so many incredible people in this room. And then went off to join I like to call it the NIH DCPFAP family, working with Dr. Mazur, Dr. Kodalil, Dr. Anita Coley, Rachel Silk, and so many other people in this room. Really as part of a team that was committed to innovative clinical research and community impact. And Sham, I'm going to say this, I know you don't really like me saying these things, but I'm going to say it. So I think, you know, Sham's, you know, scientific rigor and that belief in people, actually, and commitment to excellence opened a lot of doors for me, and not just me, but many other people who trained in his lab. And, you know, that lesson about creating space for others to lead has stayed with me and continues to shape how I mentor and lead today. So to this entire community, thank you. Your commitment to patients goes beyond this world, and that grounding, that belief that science must always serve people continues to guide my work today in Gilead, in drug development in virology. And so that finally brings me to the topic of today's theme. Viruses cannot wait, neither can we. They don't wait for our systems, our silos, our timelines. And so innovation and collaboration at purpose can't wait either. Okay. But as everyone knows in this room, innovation is extremely hard. You know, drug development is long, it's complex, it's challenging. It's also extremely resource intensive. It requires many, many years of sustained investment and collaboration. It's a field, I don't like to call it failures, defined by as much persistence and learning, by the successes as well. So it's not shaped only by the breakthroughs that we hear in the headlines, but it's everything we discover along the way. It's the biology that we don't yet fully understand. I mean, it's much easier to cure mice than people. It's the molecules, the many molecules that don't move forward, but we learn something from them. It's these safety signals that sometimes surprise us. But yet it's where the most extraordinary breakthroughs happen, because behind every therapy that finally makes it through is a patient. It's a story, and truly that's what makes the science all worth it. Okay. And you know, when we talk about innovation, it's we often think of like a single breakthrough, and it never is. It's not about one new major therapy or discovery, but innovation is really a continuum. And it starts with discovery, often in a lot of academic labs. We had dinner with some of the people in the basic science team at the IHB yesterday, often in academic or research settings, because that's where the fundamental insights actually emerge. And then comes translation, where those discoveries evolve into actionable mechanisms and targets. And then we move to development, which is where I sit, and really exciting, where the science meets the patients, where the data, the safety, and the outcomes start taking shape. But innovation never stops at approval. It continues through access, ensuring that the treatments reach those who actually need them the most. And finally, back into real world impact, because the experience in the community actually feeds right back into every single stage. And when all of these connect, that's when science truly transforms lives. So you'll see that arc play out in two stories I'm going to share today. So one is the hepatitis story, and I like to call that the quiet crisis. And then there's a COVID story, which is where science had to move at the speed of need. And I think together they reflect what's possible when science, persistence, and collaboration across all sectors together, they come together to change really what's possible for patients around the world. So I'm going to start out with hepatitis C, Anita. So, this became one of the greatest success stories in modern medicine. But as an attending in the DCPFAP clinic, this is what we had to offer our patients, Rachel, and Anita, right? So we had a year of weekly interferon injections and 20 pills a day with very brutal side effects, and which is why less than 5% of patients had ever been treated. And so we told patients then, we will try, but we cannot promise a cure, and you might not even be able to tolerate the treatments. But that began to change with some of these early clinical trials that we were involved in, evaluating a compound called PSI 7977, which eventually became sofosbuvir. So along with the work that the NIH DCPFAP team was doing, which actually really talks about the collaboration between academia and industry, there were important single center trials happening at the NIH and the DCPFAP program in the populations once considered too difficult to treat: African American genotype 1, HIV co-infection, the people who had the lowest cure rates for hepatitis C, and all these other negative predictors. And then across the globe, there are also multiple trials that were underway. Together, these efforts all showed something quite remarkable: extremely high cure rates with short, well-tolerated regimens. And in 2014, sofosbuvir was approved, ushering in this era of single tablet, one pill once a day regimens with cure rates of over 95%. Of treated individuals. So a really major milestone in medicine that actually permanently changed a field of medicine. But honestly, when we take a step back, what seemed like this overnight success about this one pill regimens, it didn't happen overnight. It was built on 40 years of science, which began in the 70s with Dr. Javi Alter's work at the NIH that characterized non-A, non-B viral hepatitis, followed by the identification and cloning of the virus by Michael Houghton and the Chiron team in 1989, and later on the development in Rockefeller by Charlie Rice and others. That really opened, the replicon system, that's what opened up the way to drug screens, targeted drug screens. And those decades of foundational science paved the way for the drug development that we could conduct in industry. But even with those discoveries in hand, I think the path to cure, and I think that's why I'm showing this slide, was anything but straightforward. So if you look from the industry lens, from the drug discovery development lens, so this is 20 years of effort before sofosbuvir was actually approved. What you can see is 712 unique compounds with an overall success rate of 2%. Most of those molecules never even reached late stage development. Only 12 things made it through of those 712. And I think that just speaks to how challenging drug development is, but requires committed resources and persistence. And then even at Gilead, so for the prior decade before 2014, that same persistence carried through. There were actually dozens, about 30 research projects at Gilead that finally identified these promising molecules that we could then later combine. And even when you look around those 4 therapies that Gilead eventually got approved, we had 483 clinical trials. Because there was just this really intense focus that we were going to look at every single population to make sure there was no patient left behind. So the result overall, as I said, changed the field of medicine, was transformative, but also a reminder that true innovation, it takes time and it's sustained investment across many sectors. And because of that collective effort across all sectors, today, there are more than 13 million people around the world treated for hepatitis C. But even with that progress, and I think we were talking about this at dinner yesterday, there's still more than 50 million people who need treatment, including 3 million here in the US. And, some of those 3 million people are in Baltimore, they're in DC. So while the science has succeeded, access and equity still lag behind. So we've seen successful national elimination programs in Georgia, in Egypt, in Iceland, but in many other places, including the US, there's still significant gaps. So, my message is eliminating hepatitis C is not, it's going to take more than great drugs. It's a holistic approach that connects innovation with access and policy and partnership across all the different sectors. And I think that sense of purpose and urgency that got us to those first few steps along the continuum have to continue to be maintained for us to actually get to hepatitis C elimination. But the story of viral hepatitis doesn't end here. For all the progress we've made in hepatitis C, I think hepatitis B and D remind us how hard this journey can be and why we can't lose momentum. So today, there are still 250 million people around the world with chronic hepatitis B. And every year, more than 1 million people actually die from complications like cirrhosis and liver cancer. Like 50% of all HCC across the world is from hepatitis B. So in the US, there are about 2 million people, but only a fraction are actually diagnosed, and even fewer are on treatment, as you can see here. So while we have effective antivirals that can suppress the virus, reverse fibrosis, reverse cirrhosis, reduce the risk of liver cancer, there's still limitations. Nucleoside therapies do not cure because the HPV integrates into the host genome, causing genomic instability and oncogenic activation. So that's why even people who are not cirrhotic can still develop liver cancer. And so the need for functional cure remains extremely important. And again, I think this slide shows us that it's not going to be easy to get there. Rasham, you spent probably 25, 30 years of your career also working on hepatitis B. I think as a field in general, we've explored almost every approach: blocking replication, silencing antigens, innate immunity, enhancing adaptive immunity, and even directly targeting viral DNA. It's decades of work, lots of approaches, but most of them have not succeeded. However, I think every attempt that we've made teaches us something new. It's about the virus, about the immune system, and what true functional cure for hepatitis B is going to require. We're not there yet, but I think, there are new modalities coming through. There are combination approaches coming through. And while some folks are exiting the hepatitis B, I do think there is renewed energy across academia and research and industry to really try and find what's possible in functional cure for hepatitis B. And so even within that larger hepatitis story sits another story, and that's the story of hepatitis Delta, which is a most severe form of viral hepatitis. So this is hepatitis D. It affects only people who live with hepatitis B. It's a satellite virus, about 8 million people globally. And for decades, it was called the forgotten virus because there are no approved therapies, even though it's known to dramatically accelerate the rate of liver disease progression. But that changed in 2020 with the approval of bulevirtide in Europe, which is the first approved therapy for this devastating disease. But, I think the important story here is that it was a breakthrough that was made possible by years of collaboration between academia and biotech innovators and industry, showing that even in rare diseases, you're able to actually create innovations that change the trajectory of care. So I think, honestly, as we strive towards the WHO goal of eliminating viral hepatitis as a public health threat in 2030, the success is going to take continued innovation, it's continued partnerships, and a collective will across all sectors, and not just in the US, but globally as well. Because I think the hepatitis story reminds us that innovations take years, but decades quite often, but sometimes the world doesn't give us 20 years. And sometimes that challenge actually arrives overnight. And that brings me to my next story, which is the story of COVID-19. Okay, so, this for us at Gilead began quietly, as most things do. So I still remember on December 31st, 2019, there was an email that came about cases of mystery pneumonia in the Wuhan district that came through from Dr. Fred Hayden, who is one of our collaborators at University of Virginia. And so I sent this email to Thomas Sillar, who's our head of virology research, and myself. And his email message said, "Florence monitoring, Happy New Year." That was the message. Tomas was in Prague, and I was in Foster City. And that message, as ordinary as it seemed, actually marked the start of one of the most extraordinary scientific mobilizations of our lifetime. So immediately for us, we began piecing together what we knew. Could this be a coronavirus given the location? What information existed? What tools did we have at Gilead that might help? And we pulled out the dossier for remdesivir, which is a broad spectrum antiviral that had been studied for filoviruses such as Ebola, and had also shown preclinical activity against other coronaviruses like SARS and MERS. And we told ourselves, well, if this turns out to be another coronavirus, maybe we can help. However, we also knew that many times with many outbreaks, by the time we actually start and the science catches up, then the wave has often passed. So for us, 1 week later, it was confirmed to the world that this was actually a new coronavirus. And then 2 weeks later, on January 25th, 2020, it was like 8 p.m. I remember because we just had dinner. I got this call from a physician in Seattle. His name was George Diaz. And he had a patient, which was the first confirmed US case of COVID-19, who was deteriorating. And he asked us then whether he could access remdesivir under compassionate use for this disease. So that evening, all teams across Gilead, the FDA, because this was compassionate use, and clinicians in Seattle worked tirelessly through the night to review the data and assess the benefit risk for this drug. And by the next morning, we'd gotten the approval from the FDA that we could try it. Consent had been obtained. Remdesivir vials were released from our San Dimas facility in California, flown to Seattle, picked up by the courier, sent to the hospital. A few hours later, infusing in the patient. And within days, the patient had improved, was discharged. The case was published in the New England Journal of Medicine. And this was the first patient that we treated with remdesivir for COVID-19. And from that moment on, the urgency, 2 days later, cases from Hoffman Estates, from Cedars-Sinai, tons of cases pouring in from France, Italy, Spain, and the number of cases grew rapidly every day. And for us across Gilead, I think what I'm most proud of is, across the entire organization, we mobilized on 3 things. 3 priorities. One was we needed to establish safety and efficacy through clinical trials to understand if remdesivir was actually beneficial for this disease. And then I think another important thing was scaling up manufacturing at risk. So remdesivir at that point had a very long, complex synthesis. It's actually more than 20 steps, some of them sequential, with specialized reagents and time-intensive purification. So ordinary to produce a single batch will take 9 to 12 months. So in January, before any of the trials had actually even started, we made the decision to scale up manufacturing at risk, because that meant repurposing production lines, securing these scarce raw materials as the global chain supply was shutting down, building new supplier networks, and recruiting partner manufacturers on several different continents, because we knew that what if this drug actually worked? The one thing you can't buy back is time. So we needed to make those decisions very early on. And I think third was access. True to who we are at Gilead, from the outset, we worked to ensure that if remdesivir was going to be, if it proved to be beneficial, it would be made available globally. So we expanded our compassionate use and managed access programs to the largest to date, and simultaneously began setting up voluntary royalty-free licensing agreements with generic manufacturers that could supply 127 low and low middle income countries, ensuring that if this actually ended up in a global pandemic, patients everywhere could be reached. And those three pillars actually guided everything that came next. And what followed was nothing short. Many people were part of this effort of a Herculean global response. And I think, for us all, that's the fastest mobilization of science, of medicine, and humanity in our lifetimes. So we were working very closely with the NIH, with academic partners, with global health agencies. We launched multiple trials in record time. Those, until the PURPOSE trials happened, those were the biggest trials at Gilead, with 10,000 plus patients. And these were happening even as hospitals were overwhelmed and travel had stopped. And the investigators, nurses, healthcare workers were caring for sick people while also simultaneously running trials. Figuring all this out, so the scientific community, all in real time and often at great personal cost. And I think all those different investigators that helped us understand the science while also taking care of sick patients are really the unsung heroes who actually made progress possible. For us, this was new territory. We're all learning together. Academia was learning, government was learning, industry was learning. But I think every step of the COVID response was anchored in science and on the patient with urgency and compassion, realizing that every day you could get to the story faster for us at Gilead was a day that meant someone for someone's mom or dad, and often our colleagues as well, who had COVID. So by October 2020, less than 9 months after that initial email, remdesivir became the first FDA-approved treatment for COVID-19. And it was a historic moment, not because of one company, but because of what collective determination and purpose can make possible. And so, what all this work meant was at the height of the pandemic, remdesivir had actually reached millions of patients. Until today, it's treated over 15 million people worldwide, and more than 60% of them are in low and middle income countries. So for us all, it was an unprecedented response to an unprecedented pandemic, the worst in 105 years, the worst in our lifetimes, and was only made possible because of collaboration, from industry, from academia, from government, and public health institutions worldwide. And so, while, again, the COVID response unfolded at this breakneck speed that we could all see, the reason it could happen actually was because of the decade behind the remdesivir story. It didn't start when remdesivir got approved. It wasn't developed overnight. It was built on more than 10 years of foundational research and a commitment that we made to the field of emerging viruses long before anyone had heard about SARS-CoV-2. Through multiple partnerships, we had conducted a lot of in vitro and in vivo work and studies that demonstrated remdesivir's broad spectrum activity against multiple viral families, including coronaviruses. So that groundwork and the scientific infrastructure built around it is why when COVID hit, we weren't starting from zero. And I think that's a lesson we all must carry forward. Preparedness cannot be built in the middle of a crisis. You have to actually prepare for it. You can't do that in the middle of a crisis because preparedness isn't a moment in time. It's a mindset that you actually have to have. And so for us, that same mindset continues to guide the work we do today. So in the past few years, we've been part of, different outbreak responses from Ebola, Sudan to the Marburg virus in Rwanda. And our goal is to be better prepared than the last time. And we're trying to do that also with new oral therapies that we're developing that could be deployed faster and used even earlier for, post-exposure prophylaxis, which is the driver behind, obeldesivir and some of these other investigational agents I'm showing here. We're also working across board with academia and global health organizations to ensure that we're also studying combination approaches, pairing antivirals with monoclonal antibodies to see if we can enhance efficacy against filoviruses, which have some of the worst mortality rates. And we're working closely with all these different organizations as part of a much broader effort, such that we can ensure that when the next outbreak occurs, that we're not just reacting, but we're actually ready. Because I think for me, the truth is it's not a matter of if there'll be another outbreak or a pandemic, it's when, right? In the last 20 years, we've seen Zika, Ebola, SARS, MERS, COVID-19, monkeypox, each reminding us that viral threats continue to happen and they're increasing in frequency and complexity. And that's why we need sustained research and strong partnerships and a global mindset of readiness built quietly year after year, with an understanding that even if you don't know what that next virus is, there are things that you could do to be ready when it actually comes. And so that's why this slide for me means so much. This is a remdesivir story. Doesn't rest on just Gilead or a single company. It takes alliances between academia and industry and NGOs and governments working together to build the networks, the protocols, the partnerships, so that when that next threat emerges, we are able to respond together and faster. Because in the end, it's not a science story. It never is. It's a human story. Every clinical study, every data set, every process improvement comes down to one thing. It's people. Because behind every study is a name. It's every breakthrough is a life change, whether it's Nurse Colleen who survived Ebola, or the newborn baby, or the families reunited after COVID. And for us at Gilead, that's really what keeps us moving. And so as I reflect on the story of hepatitis, of COVID and beyond, I am always reminded that innovation doesn't happen in isolation. So at Gilead, we have a deep commitment to science and patients, and that has defined our story for over 3 decades. For many years, we've been at the forefront of tackling some of the world's greatest public health challenges, whether it's HIV, to viral hepatitis, to COVID-19, to emerging viruses. And we're also now bringing that same focus to the work we're doing in oncology and inflammation, because our mission has never changed. It's been the same: to discover, develop, and deliver. That piece is so important. Transformative therapies that improve the lives of patients across the world. And what defines that legacy is not just the science. It's all the different partnerships I showed you that make it possible across academia, government, industry, global health, and that's how we keep moving forward together. And so for me, finally, the lessons I learned early in my career at Maryland and at the NIH are the same ones that continue to guide me today. Science is only as powerful as the purpose behind it. No patient left behind, as he said, Dr. Mazur. Those same principles guide me at Gilead with what I do today in discovery and development or delivery of transformative therapies to serve patients around the world. The setting may have changed, but the purpose is still the same. And we have to use science to make a difference in people's lives. And so that brings me full circle. When all these different levers of discovery and translation and development and access and real world impact come together, we actually don't just make medicines. We actually change what's possible for patients. And that's what will prepare us for the future, because viruses don't wait and neither can we. So with that, it's my great pleasure to hand over to my friend and colleague, Dr. Moupall Das, who will share an exciting story of innovation that's transforming what's possible in HIV prevention and beyond toward a future where we can actually someday end the HIV epidemic for everyone, everywhere. Thank you.
Thank you so much, and even though you tried to steal my job. So it's now my pleasure to introduce Dr. Moupall Das, who's the Vice President of Clinical Development of HIV Prevention and Virology Pediatrics at Gilead Sciences and the head of HIV Prevention. So Dr. Das completed her AB in Biochemical Sciences at Harvard College, followed by medical school and residency in internal medicine at Columbia University and New York Presbyterian Hospital. She subsequently completed fellowship in infectious diseases at the University of California, San Francisco, as well as a master's in public health and epidemiology at UC Berkeley. After completing her training, Dr. Das worked at the San Francisco Department of Public Health as Director of Implementation Science and Evaluation Research, and at UCSF, where she cared for patients in the Ward 86 clinic. During this time, she developed the concept of the community viral load, a novel population-based indicator to evaluate the impact of treatment as prevention. Her research was the basis for measuring community viral load and using viral suppression to evaluate the effectiveness of President Barack Obama's national HIV/AIDS strategy. She also served on the Institute of Medicine Committee on Data Systems for Monitoring HIV/AIDS Care. Since joining Gilead Sciences in 2013, Dr. Das has leveraged her clinical HIV and ID expertise, clinical trial design and epidemiology skills, and passion for coaching and mentoring to build and lead high-performing cross-functional teams in HIV treatment and prevention. For the past approximately 5 years, Dr. Das has led the PURPOSE program, leading to the groundbreaking development and approval of Lenacapavir for HIV prevention, a drug that has been projected to revolutionize the fight against HIV. Perhaps most notably, she has maintained a consistent and passionate commitment to health equity, designing trials that are inclusive of people who inject drugs, transgender individuals, adolescents, and pregnant women, populations frequently excluded from research. In addition, she has consistently ensured input and feedback from community members with lived experience throughout the study design and implementation process. For her groundbreaking work, Dr. Das received the 2025 Manny L. Baumick Breakthrough of the Year Prize on behalf of Gilead Sciences and alongside collaborators working on Lenacapavir. And today we are incredibly honored to have Dr. Das with us as the 18th speaker of the IHB Annual Greenebaum Lecture.
Lower it from Anu and everybody else. Hello, everybody. It's so good to see friendly faces in the audience. And I was saying yesterday and this morning that I feel like I'm crashing on Anu's like work reunion and training reunion, but it's great to have the opportunity to talk to you guys about the PURPOSE program, which for the since 2018 has been my purpose. So let's get into it. This is a picture I took when I was working with Paul Farmer during medical school, actually. And we were working, I was helping him, do the references for his book, Infections and Inequality. And my favorite quote from the book is, "Infections track along the fault lines of social inequality." And one of the things that I've learned throughout my time in public health, academic medicine training, and especially in my time at Gilead, is this corollary that I think is really critical for this statement, which is innovation without intention can unwittingly exacerbate disparities. We've had a huge commitment over time, and you've seen another version of this slide from Anu, in developing HIV treatment and prevention medications, and you can see all the programs that we've worked on and several of our firsts at Gilead. And the number of people, over 16 million people, have been accessed a Gilead treatment or PrEP regimen, including many in low middle income countries. But there remains a significant unmet need for more options for HIV prevention. Everyone in this room knows the statistics that over 1.3 million new cases of HIV globally, only 36% of the 1.2 to 1.6 million people who would benefit from PrEP in the United States are on it, and there's significant disparities in who is accessing current PrEP regimens. And globally, it's much worse, with only 8% of people who should be on PrEP. Why is this happening? There's many reasons, including a low perception of PrEP benefit, adherence challenges, also demands on the healthcare system and challenges with access, and all the isms in terms of systemic sociostructural factors. We think the solution is innovation in both science and in health equity. One can't happen without the other if we're going to truly try to end the epidemic for everyone, everywhere. So we are committed to evaluating new drugs including long-acting twice-yearly subcutaneous lenacapavir. I have to update the slide. It's no longer investigational, as well as using innovative trial designs to help answer questions that could not be answered before the counterfactual design. But we're doing this also with a focus on innovation and health equity, building on partnerships that we've had over years, really truly representing the voice of the people who would most benefit from PrEP, including working with community members and establishing our own global community advisory groups, a focus on person-centric design in the trials, which will yield a focus, hopefully, on person-centered access and implementation, and of course, making sure that the most disproportionately affected people are included in the design, conduct, and implementation of our trials, because unless we pay attention to diversity, equity, and inclusion, we're not going to be able to end the HIV epidemic. So this persistent commitment that Anu talked about led to lenacapavir. And our timeline starts in 2006. However, the Wes Sundquist and his team at University of Utah and several people were working on delineating capsid before 2006. But I just want to call your attention to the top left. Though each dot is a molecule that we studied. And maybe in the future with AI, we won't have to study every one of these molecules in the lab, but that's what it took to get from getting from the least desirable to the most desirable in the bottom right there over the 6-year period of time. And that is the second generation lead. So bear with me. So a capsid is a fullerene clone. That's just interesting if you like math. But the cool part of capsid is it's a beautiful geometrically designed thing of all of these monomers that get together in hexamers and pentamers to form this beautiful geometric structure. And initially we thought that we could get an inhibitor in there to slow down the development of this beautiful geometric structure. That was our first lead, but that didn't work. And then someone really brilliant had the idea, why don't we actually come up with an accelerator, something that speeds up assembly? And that's what we did. It speeds up assembly, but makes it defective. So that is the way lenacapavir works. And I didn't really understand that until a couple of years ago. Anyway, you can see the very rapid progress of lenacapavir, both for treatment of multi-drug resistant HIV in people who are heavily treatment experienced, along with a background optimized regimen for HIV treatment, as well as our recent US and global approvals for Yeztugo, and I'll talk about that in more detail in a few minutes. So what is lenacapavir? Here's the picture. It's not, can't really, it's much more cute in 3D, I promise. But anyway, lenacapavir is a small molecule with exceptional potency, high stability. It has a multi-step mechanism shown on the bottom left, and it's really the backbone of our future treatments in treatment and prevention. And we at Gilead are really committed on taking our current options and making breakthrough medicines and ultimately game changers. And we do this with many of the pieces in HIV are exactly the same as what Anu described in hepatitis and in COVID, with multiple approaches, innovation in our medical chemistry and formulation, and expertise in virology across all functions, and really sharing lessons learned. So twice yearly lenacapavir has been approved for as Yeztugo and Yeztugo in Europe, and we're studying once yearly lenacapavir, which took us a very long time to develop, but we made it work. And we're really excited about that trial, which I'll also tell you about in a second. So I like to use this beautiful acacia tree to talk about our program, because our program, lenacapavir, and the 5 trials, Purpose 1, 2, 3, 4, and 5, really built on the foundation of what Martin Ree and his colleagues did for HIV treatment for MDR HIV, as well as proof of concept in non-human primates. And with that as our foundation, and then the roots are really our 4 pillars of purpose, health, equity that I described, and that yielded purpose 1, which was our trial in cisgender adolescent girls and young women in South Africa and Uganda, purpose 2, which was our trial in cis, gay, and other men who have sex with men, trans women, trans men, and gender binary individuals in the United States, South Africa, Peru, Brazil, Mexico, Argentina, and Thailand. And this was the first trial to include intentionally transgender men and gender non-binary people, which we're really delighted about. And then we are collaborating with NIH and the HIV Prevention Trials Network on Purpose 3 and Purpose 4, which is HPTN 102 and 103. We couldn't get those numbers to line up. And Purpose 3 is in US cisgender women who could not be included in Purpose 1, really focusing on Black women and women of color who are disproportionately affected by HIV in the US. And Purpose 4 is our study in US people who inject drugs. We're also doing Purpose 5 in France and the UK to look at the relative benefit of long-acting lenacapavir for persistence over oral therapies. And we started in 2019, and I love this quote from ACT UP back in the day, "Nothing about us without us." And when we started this trial, we knew we wanted to do something differently. We knew that pregnant women, lactating women were disproportionately affected by HIV, and we wanted to include pregnant women. We also knew that young people were disproportionately affected by HIV, and we wanted to include young people in our clinical trials, and we did. But this was not easy. It required changing hearts and minds internally with our safety people and our legal people and all the people who think about risk. And it also required changing hearts and minds of regulators and ethics boards and some investigators who took more of a let's justify inclusion approach rather than let's justify exclusion, because we wanted to protect pregnant women through research rather than from research. But what helped us the most was when we went to Kigali in 2019 and held this first community meeting. It was a multi-sectoral meeting of civil society. You'll see one of the directors of health of the public health system in Rwanda on the slide on the upper right-hand corner, as well as many, many people who are investigators, site staff, community advocates and activists living with HIV, and also those taking PrEP. And there was a young woman, 17 years old, who, after listening to all this debate about whether we should include pregnant people or not, stood up and revealed she was quite pregnant. And she said, "You need to include people like me." She revealed she was taking Truvada because she didn't want her or her baby to get infected with HIV. And she said, "We have to include people like her and her girlfriends, people who are pregnant and young people in our trials." And I always use her story because you can have all the data you want, but you need a good story to really change hearts and minds. And that was incredibly helpful because it was very hard to get this off the ground. These are some of the other things that people told us when we established our global community advisory and accountability groups, one for each trial, with representation of all the populations that are disproportionately affected, that we wanted to prioritize inclusion. Those were the folks in the community advisory groups, and we did all the things that they asked us to do. Here's the Purpose 1 trial design. We can talk about the counterfactual if anybody cares later. Here's the Purpose 2 design. So Purpose 1 evaluated both lenacapavir and Descovy, or FTAF, for efficacy in women. Purpose 2 evaluated lenacapavir because Descovy had already demonstrated to have efficacy in this population. And here are the locations where the trial took place. It was the most globally, racially, ethnically, and gender diverse phase 3 HIV program conducted to date. 67% non-white, 63% Hispanic and Latina, and 22% gender diverse. In contrast, Discover was less than 1% gender diverse, which was a trial of Descovy and Truvada in gay men and trans women. Now, the status is that over 95% of the people after the primary endpoint have been chosen to continue into our open label extension, where they're receiving lenacapavir injections in all 4 sites that we are studying. And participants will continue to receive len through the trial until access is available in their location. So how do we pull this off? Again, a combination of innovation in science and in health equity. Really requires an ecosystem of all people working together from their angles, whether it's investigators, the community, our CROs, our internal teams, from our regulatory people and our operations people to our manufacturing people, our clinical pharmacology colleagues. And without this joint innovation in science and health equity, we would not have been able to achieve the unprecedented efficacy seen in the trials, nor would we have been able to achieve the unprecedented inclusion in the trials. So we're very proud of these firsts in the Purpose 1 program. First, to include pregnant and lactating people. First, to include adolescents, and following the guidance and the advocacy from investigators, providers, and patients alike. And including these folks avoided the usual 8 to 10 year delay in having pregnancy information about antiretrovirals in labels and for guidelines. And I just wanted to highlight, this is a picture of where in Uganda we had one of our clinical trial sites. And with permission, these are the Purpose moms and babies from that Uganda site. But I wanted to show you how these women arrive on these canoes across Lake Victoria and where they deliver. People will not deliver pregnant women in these sites because there's a dearth of supplies. So people were arriving in labor and not being able to be attended to because the places didn't have enough supplies. So we started providing the mama kit and supplies out of the requests from the investigators to ensure that we could optimize safety of our participants. And another tiny story I'll tell, there's a lot of pressure on our teams to not do anything that would be considered undue incitement to participation in clinical trials. So these girls were coming from Kalangala and these islands on the lake over to the mainland, and our study visits take a long time. Like, you know, you have to wait for labs, there's multiple things, there's PROs to fill out, and we wanted the girls to be able to stay overnight at a hotel on the mainland. And one of our internal colleagues who was reviewing this request asked us, why can't they Uber home? And we were like, let me send you some photos. And having these photos and this video of them like using a machete to cut through the grass to help set up this site was very informative. And it's really hard for people who've never left the United States and who don't really understand the difference between trying to do a trial in Uganda and doing something in San Francisco, where we could certainly have paid for an Uber for people to go home. Anyway, we're very excited about the inclusion in Purpose 2. We were able to follow the guidance and asks from the community with the No Data, No More Transgender Manifesto. And of course, Purpose 2 also included adolescents with a focus on recruiting 16 through 25-year-olds. And the intentional inclusion of these vulnerable key populations globally helps really support earlier access for these disproportionately affected populations. So I talked about the 10-year delay and how we avoided that by having data in pregnancy, lactation, and young people, and having truly one of the most inclusive indications with our first label. Now let's talk about what we're doing for the rest of the world. So we're very pleased to share that we are approved in 4 locations. So the US, EMA, South Africa, and Zambia last week. The Zambia approval took 17 days. That's never happened. Yes, I do think that requires a round of applause. Our team, thank you. Thank you. Our team cross-functionally, this takes hundreds of people internally. I like to say that a filing is like 5 to 10 R1s and a whole bunch of other stuff, like doing R1s for manufacturing as well as for the science. After we got the CHMP positive opinion, that's the Committee for Health and Medicinal, anyway, it's part of the EU, and they do this initial positive opinion, and then you get the approval. So we got the positive opinion in July and the approval in August. But that positive approval led the way for the WHO pre-qualification process, which we received in October. And we worked with multilateral collaborators to figure out what the priority countries should be. So PEPFAR, USAID last year, as well as several other multilateral organizations, UNITAID, Gates, others, to figure out where this would be implemented. So the countries that we filed in first would be the countries that would be receiving product through all these global mechanisms. So these are our WHO CRP priority countries based on all of that discussion, which took place for years prior to our results. We've also filed in these other locations, and these are the countries, the CRP countries that we've already filed in. And of course, we have approval in South Africa and Uganda. And lastly, this is what the team has to do by the end of the year. These are the rest of the countries. They have to apply and they have to finish the CRP list. So I'm checking my email. I have to update this slide literally like every day because the team files in another country and it makes me so happy. So I would just say you can have this, the innovation in the science and the innovation in health equity, but regulatory approvals alone are not enough to ensure access. And we saw this with the first approvals of Truvada in 2012 for PrEP and how slow that was to roll out globally. So in order to support access in low and low middle income countries, these are all the things that we've done. So voluntary licensing agreements to generic companies. We shared our IP even before we filed with the FDA. The minute we saw the results for Purpose 2 in September of 2024, we started sharing our IP with the generics that we chose. We shared how to make the products. We have manufacturer bridging supply because we know even when generics get it and they start making it, it takes them like 2 to 3 years to get up and running. So we have manufactured bridging supply at risk for 2 million people for 3 years to get Lenacapavir while the generics get on board. We're doing the simultaneous global filings. I've been very closely working with CDC, WHO, and others on the guidelines and pre-qualification and all the multilateral agreements that I mentioned. Here's a bit about that timeline and how rapidly this went. This is really, really unprecedented. And we have Purpose 3, 4, and 5 that I told you about. We'll have data there in the middle of next year and be sharing that in the end of next year and early 2027. And we've also been focusing on using implementation science to increase access and uptake. And so we have the PROPEL study. So we're going from Purpose to PROPEL. And we also are working with various other implementation studies in the US, as well as with UNITAID on IMPREP, LEN, and Project PrEP, and align with the Gates Foundation. And very important, if you're interested in working with us on the on research in the PEPFAR priority countries, we'll be issuing an inclusion global RFP later this year. I hear it might be in the beginning of December on an important date then, but don't hold me to that. So keep your eye on our website for those RFPs. And I mentioned that we don't stop. So we're moving forward with focusing on long-acting orals and our once yearly injectable. This took a long time, and I was there for every formulation, and we had 30 or 40 different formulations trying to figure out how to get Len to last for a year, but the team did it. And we enrolled our first participant in July of 2025. We expect results in the middle, towards the end, I would say in 27, and hopefully heading to approval after 28 if all things go well. And I'd just like to say our tree is becoming a forest. So here's our implementation science tree. Here's Purpose 365, which is a smaller trial that's going to harness all the data from before. And if you want to learn more about access, scan this QR code. It is an updated website on the Gilead website that talks about all of our efforts to make sure that Lenacapavir truly can help end the epidemic for everyone, everywhere. So thank you so much and for the opportunity to tell the story.
Thank you so much to Dr. Das and Dr. Osinusi. We now have a little bit of time for questions if anyone in the audience has anything they'd like to ask these two amazing women. Team Gladwin.
Yeah, thank you. Yes, so that drug actually is it's quite similar to remdesivir. It's actually the same mechanism of action, but I think what we recognize, remdesivir has been around for a few years, but it's intravenous. And as we've continued to work with the different places where the outbreaks happen, yes, I mean, when people are sick, you can get them an infusion and they have GI bleeding. So maybe they do need intravenous agents, but we needed oral agents because that's how you're actually able to help with post-exposure prophylaxis, whether it's the ring around with the healthcare workers as they are doing vaccinations as well. And so it's a very similar drug to remdesivir. There's obeldesivir, and then we're working through a few other antivirals as well that we could be able to deploy orally instead of remdesivir. And yes, it is in the yes, it's a nucleotide. Yes. Dr. Mazur?
No, actually, it doesn't seem to be a logical part of the portfolio. Why is that part of your portfolio if you're a company that is driven by related to are you taking part of the company? Why is it?
Well, I think we'll do a too far here. I get your question, Dr. Mazur, and I think you actually know the answer to that because I remember when I was, thinking about like moving to industry, and you're like, you were supportive of Gilead for some reasons because of the focus has always been around science and the patients, and I think true to what Gilead has been for the 30 years, and maybe it's because it started out as a virology company, recognizing that for us, even on a daily basis, the discovery and development piece is critical. We spend a lot of resources doing that, but the delivery piece it's like three pillars. They're equally important because there's no point developing drugs if it's not going to reach the people who actually need it. And so I think it's just all part of the DNA at Gilead of making sure that whatever we're working through and partnering, because we recognize like we're not going to do all these things alone. We have a lot of amazing partners in the field, in academia, in governments, in public health institutions who are actually the ones on the front line making sure this happen. And any way that we're able to actually facilitate that and make that happen, I think is critically important to who we are, but Moupall probably has a better answer.
No, I would just say, yes, we're a publicly traded company and we have to keep our investors and Wall Street happy. But I think our goal historically has always been to two things: doing well to make the investors happy and doing good. And in terms of HIV prevention and all of the viruses that we talk about, we can do well by doing good. So that's our goal. Yeah.
Thank you for the funding to this company.
Right, so I'll take this first. So I'm sorry, I thought this was a mic, but it's not, it's something else. I'm usually loud enough to be heard. Yeah, so again, we're a pharmaceutical company that's publicly traded, so we're not a funding institution. However, we do a lot of studies. When I first joined Gilead, I didn't realize that many of the investigators who I thought were purely NIH funded actually had grants from Gilead. So we do investigator-sponsored research where somebody comes up with an idea and engages with our medical and commercial teams and proposes that idea to Gilead. We also do requests for proposals. So the RFP that I was mentioning that's going to come out in December is for implementation science in the countries where Len is being implemented globally by multilateral partners. And in addition, we, obviously we are impacted because all of our sites are funded by different government agencies or internationally through Global Fund and PEPFAR and CDC and others. So there has been an impact at our sites and we are doing what we can. So we've obviously increased funding to the sites so we can safely conduct our clinical trials globally. And at the end of the day, it's an ecosystem and other countries and other places are stepping up, like the Nordics, like the UK, like South Africa leading the replenishment. Like there's a lot of countries and other players who need to come and fill the void that have been made by changes in the last year. But I'm incredibly, incredibly optimistic about getting Len to the people. If you just Google Lenacapavir in South Africa, you can see the enthusiasm and the approval and their plans for access as early as next year.
Maybe just to add on to what Moupall said, you're absolutely right. I think we all feel it wherever you lie in the health continuum, whether you're in academia or the government agencies or the public health institutions that we work with, the big ones, or industry, because it's, as Moupall said, it's a big ecosystem that needs to continue, the continued partnerships are critical. Science doesn't stop, patients don't wait. Like we have to keep pushing ahead. And I think in our part, we're committed to doing whatever we can. And I think a good thing that one piece you said is we fund hundreds of these investigator-sponsored research programs, as well as collaborative research as well, right? Because when I said that 483 clinical trials, a lot of those were collaborative research as well. Because if we're trying to get to the answer of no patient left behind, we have to go beyond those initial trials and really make sure that we're working through understanding all the different patient populations. And then, as I said, that access piece is so important. So a lot of what we also do now is funding like some of the implementation science work as well. But we know this last year has been quite challenging, but hopefully the collective will to understand that science needs to continue to be funded will happen. Oh, sorry, there's someone at the back.
Yes. Right. So I didn't really talk about treatment in the talk, but so lenacapavir is approved as Sunlenca for the treatment of multi-drug resistant HIV. And we're currently studying lenacapavir and bictegravir together, Biclen as a daily oral pill. We're also partnering with Merck to study islatravir with lenacapavir as a weekly treatment. And those are the orals, so daily and long-acting weekly. We're also trying to get to long-acting monthly in both treatment and prevention, as well as longer-acting injectables. So we're also we need a dance partner for Len because we haven't found anything else the last 6 months, but we have looked at formulations of Len that are shorter acting to try to find an appropriate dance partner so we can have two highly effective HIV treatments together in an injectable regimen. And we were the first to demonstrate a 6-month regimen. So we have a current 6-month regimen for treatment that's going into phase 3 trials, which is lenacapavir with 2 different broadly neutralizing antibodies.
Lenacapavir is such a transformative. I mean, it's nothing like sort of an HIV vaccine. I don't, nothing on the rise of this path. So, working in here in Baltimore, got landscape, I see a tremendous challenge. Access. I'm wondering from your perspective, what would you like to see as policy or big policy, but funding, or how that will go? How should the state step into this field to really allow access? I just think we're gonna fall behind here, but way behind.
Yeah.
Frightens me to see that you I don't feel any problem. You try to deliver these medications that is their vaccine, or like vaccine.
Yeah. Well, it's a sticky wicket to talk about vaccines in any shape or format. I guess what I would say is our commercial and medical teams have been incredibly successful. Like, I don't practice medicine anymore, but we had a J code within several months of Lenacapavir being approved in the United States. We're at like 70%. Where are my folks? 70% lives covered. So insurance is covering it. I was just in Connecticut at Yale last week. Yale Medicaid is covering, like Connecticut Medicaid is covering it. It's on Medi-Cal in California. I don't know if it's on Medi-Cal here. And yes, it's on Medi-Cal in Baltimore. So there should be really no limitations from public and most private insurances. Like, I think we have to just go back to basics, which is expand testing and diagnosis. If people are positive, make sure they're connected to durable virologic suppressing and quality HIV care. If they're negative, offer them HIV prevention and offer them the choice of what they want that works in their lives, and then make sure people persist in care. And all that can be done with the current, with existing resources. And there's lots of places. When I was in New Haven last week, I was fortunate to see my first commercial injection of Lenacapavir, and it was on a mobile van on the wrong side of the train tracks with Dr. Sandra Springer's project that takes care of people who inject drugs. And it was a Black cis woman who got her first injection, her first prep with twice yearly Lenacapavir. And they had amazing resources on the mobile van. So these are things that we know. We've used them internationally. We've used them in the United States, and we can just build on that 30 years of experience in trying to make sure that Len gets to the last mile. Because the lower hanging fruit, there are people who are doing great with daily oral prep and current regimens. We need to get to the people who are the disproportionately affected by HIV, and it requires things like mobile vans, being out in the community, making sure that we are working with the people who are disproportionately affected, and they're helping us lead access to their people. Yeah. Right. So we've been fortunate to present our qualitative research at IS in Kigali from Purpose One. So we presented, and earlier, I can't remember all the conferences, but we did a deep dive on pregnancy and learned about people's experience while they were blinded on their Lenacapavir versus oral pills and the preference for persistent protection. There's also, we did quantitative PROs, or participant reported outcomes, in all the studies. So we have numbers on everybody in the quantitative, and then we have the qualitative data. And I believe that the Purpose 2 qualitative data will be presented in an upcoming conference, and we continue to have Purpose 1 publications and presentations. It's been very helpful for informing access and how to talk to people, and the posters have these great quotes from the participants. And if you're interested in any of our posters that haven't been published yet, although I am harassing the team to get the publications out, if you go to Ask Medical Gilead conferences, it will have every conference presentation we've given that you can download as a PDF.
The purpose trial also.
So great question. We have presented a lot of information about the lack of DDIs with, for example, statins and how to administer lenacapavir. So lenacapavir is a moderate CYP3A inhibitor. It's probably actually weak to moderate, but per label, it's a moderate CYP3A inhibitor. So it can elevate levels of drugs that are processed through CYP3A. But you can give lenacapavir with statins. You can give it with Viagra and other PDE5 inhibitors. It can be safely administered with gender-affirming hormone therapy and hormonal contraception. And there's very clear guidance in the Sunlenca and lenacapavir Yes2Go, Yay2O labels about how to administer it. And I'm actually going to go through the data today at 4 at Hopkins. So if people are interested, I can send you the Zoom. Yeah.
Aside from outbreaks.
Yeah. So recently I heard Dr. Wool say that we need to disaggregate HIV testing from STI testing. And I think he's absolutely right, David Wool. Because STI testing is determined by your epidemiology and behaviors, right? So some people need STI testing every 3 months. But with lenacapavir, you really only need to test for HIV before you give the next injection, because right? So you can reduce the HIV testing frequency, and that can save money and visits. Some people are uncomfortable with that because they want to keep seeing people every 3 months and engage with them, but they can for STI testing, and conversation doesn't need to include necessarily. So there are ways with new innovation to also innovate in reducing cost and therefore increasing access. I think it's time for us to go. Thank you.
Thank you all. A great lecture. We really expected to hear the advances in this. We have collaborated both of them, and these topics are very relevant for us at the research front as well as far as practical aspects that many of the questions came up. Thank you so much. We have, I understand that we have very busy schedule coming and accommodating a couple of days with us. It's great. And we're just going to give you an appreciation of our Greenebaum lectureship. Please accept this as our thanks for all the great work you do.
I gotta pick it up.